Hi everyone, welcome back to my A-Level Biology lecture series. In this lesson, we will explore test cross. At the end of this lesson, you will be able to interpret and construct genetic diagram, including Punnett square, to explain and predict the results of test crosses. Let's get started. So, what is test cross? So a test cross is a genetic cross used to determine whether an individual display a dominant phenotype, whether it has a homozygous dominant or heterozygous for that trait. The individual is crossed with another organism that is homozygous recessive, which allow the detection of the hidden recessive allele in the unknown genotype. So the logic of the test cross, so if the unknown individual is homozygous dominant, then all offspring will display the dominant phenotype, right? Because homozygous dominant cross with homozygous recessive during the test cross, all offspring will display the dominant trait or dominant phenotype. However, if the individual is heterozygous, the cross will produce offspring in 1-to-1 phenotypic ratio, where dominant to recessive is 1-to-1. Because why? In heterozygous individual, they have both type of allele, and when these two allele cross with the homozygous recessive, definitely it will produce a result of 1-to-1 phenotypic ratio. So this method works because recessive allele are only expressed phenotypically when in the homozygous state, make them easy to identify when crossed with a recessive. So no point we cross it with a homozygous dominant, because homozygous dominant individual will mask, the homozygous allele will mask the effect of the recessive allele. Let us start with the monohybrid test cross. So a monohybrid test cross is used to determine whether an individual shows a dominant phenotype is homozygous dominant or heterozygous when we look at one particular characteristic. For example, in garden pea plants, the seed color is determined by dominant allele Y, code for yellow seed, and recessive allele Y, code for green seed. So a plant with yellow seed may have genotype of homozygous dominant Y, or heterozygous dominant Y with recessive Y. So in order to determine which genotype it carries, so the plant is crossed with a homozygous recessive Y plant in a test cross. So let us look at the test cross outcome for seed color in garden pea plants. So if the test cross result, the offspring phenotype show all offspring have yellow seed, what is the conclusion? Yes, all offspring have yellow seed. Therefore, the yellow seeded parent must be homozygous dominant, because the parent can only produce gamete carrying dominant allele Y. So when crossed with a homozygous recessive plant, all offspring inherits Y or dominant allele Y from the yellow seed plant parents and recessive Y from the green seed parent, giving all offspring with the heterozygous, and in heterozygous case here, dominant allele Y is expressed. Therefore, all offspring have yellow seed. On the other hand, if the offspring phenotype shows 1-to-1 ratio of yellow to green seed, therefore the yellow seeded parent must be heterozygous. So the parent produces two types of gametes, dominant Y and recessive Y. So when crossed with a homozygous recessive plant, half of the offspring inherits the dominant allele Y from the yellow seed plant, producing the genotype heterozygous dominant Y recessive Y, which give rise to the yellow seed. On the other hand, another half inherit the recessive allele Y from yellow seeded plant or parents, producing a genotype recessive Y, which code for green seed. So therefore, this result in the 1-to-1 phenotypic ratio of yellow to green seed. So overall, we can see that based on the offspring phenotypes, we will be able to conclude what is the parental genotypes, okay? But again, we don't carry out test cross for recessive trait because when it comes to the recessive trait, we do not need to carry out test cross. We know their genotype automatically. Next, let us look at dihybrid test cross. So a dihybrid test cross is a genotype or genetic cross between an individual showing the dominant phenotype for two traits, but with unknown genotypes, and an individual homozygous recessive for both traits. So basically, we choose homozygous recessive because we want the recessive allele to show their effects. So it reveals the genotype of the dominant type parents by analyzing the phenotypic ratio of the offspring. So again, in the garden pea plants, the seed color and the seed shape are each controlled by a separate gene with two alleles. Seed color, dominant allele Y code for yellow seed, recessive allele Y code for green seed. Seed shape gene has dominant allele R code for the round seed, and the recessive allele R code for wrinkled seed. A yellow round seed plant could have one of the following genotypes: homozygous dominant for Y and homozygous dominant for R, heterozygous for Y but homozygous for R, homozygous dominant for Y but heterozygous for R, or heterozygous for both gene loci. So to identify the exact genotype, a dihybrid test cross is carried out. So in this case, the yellow round seed plant is crossed with a homozygous recessive green wrinkled seed plant because we want those recessive alleles to be expressed in the offspring phenotype. Let us look at the test cross outcome for seed color and the seed shape in garden pea plants. If all offspring are yellow round seed, the conclusion is the yellow round seed parent is homozygous dominant for both gene loci. Because the plant is homozygous dominant, therefore its gametes carry the dominant Y and the dominant R allele combination. So when crossed with homozygous recessive plants, all offspring have the genotype heterozygous for Y and heterozygous for R. Therefore, the offspring display the dominant phenotype, which is yellow round seed. If the offspring is in a ratio of 1-to-1 of yellow round seed and green round seed, so the conclusion is the yellow round seed parent has the genotype heterozygous for Y, but homozygous dominant for R. So the plant is heterozygous for seed color and homozygous dominant for seed shape. So therefore, all offspring shows or have round seed. So it produces two types of gametes. Half of the gamete carry dominant allele Y, another half carry recessive allele Y, but all gametes carry dominant allele R. So when crossed with a homozygous recessive plant, this results in a 1-to-1 phenotypic ratio of yellow round to green round seed. Okay, why? Because all gametes carry dominant allele R, which code for round seed. The next outcome might have the offspring in a 1-to-1 ratio of yellow round seed to yellow wrinkled seed. So in this case, the conclusion is the yellow round seed parent must have a genotype of homozygous dominant Y, but heterozygous for R. So again, the plant is homozygous dominant for seed color, yellow, and heterozygous for seed shape. Okay, so with two types of allele, dominant R and recessive R. So this parent produces two types of gametes, half carrying dominant R and another half carrying recessive R, but all gametes carry dominant allele Y. So when crossed with homozygous recessive plant, this results in a 1-to-1 phenotypic ratio of yellow round to yellow ringed seed. So based on this result, we know that yellow in all offspring definitely indicates that the parental genotype must have homozygous dominant for allele Y, but heterozygous for allele R. If the offspring phenotypic ratio is 1-to-1 to 1-to-1 ratio, so the conclusion is the parents must be, or yellow seed parent must be heterozygous for both gene loci. Therefore, the offspring show these four types of phenotypes. So the yellow round seeded plant is heterozygous for both traits, producing four types of gamete in equal proportion according to independent assortments that take place during metaphase 1. So when crossed with homozygous recessive plant, each gamete combination occur with equal probability, resulting in 1-to-1 to 1 phenotypic ratio of yellow round, yellow ringed, green round, and green ringed seed. So again, based on the offspring phenotypic ratio, we will be able to make a conclusion what is the parental genotype when this parent shows the dominant trait. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.